So first of all, I would like to thank Cure Cancer for funding my research. And I'm going to talk about today cytokine-induced killer cells for the treatment of metastatic bowel cancer. So, as you guys have heard already, bowel cancer or colorectal cancer is the second deadliest cancer in Australia and also around the world. Around 1 in 14 people, Australians essentially, will get diagnosed with bowel cancer each year. I think what hasn't been mentioned as much, though, is like if found early, the success rate for treatment for bowel cancer is quite high. It's around 99%. But currently, with how patients are getting diagnosed, a lot of them, more than around 50%, actually are getting detected with advanced stage bowel cancer. And that's the type of cancer that we are interested in. So for people who don't know, the Basel Head Cell Institute is actually our research institute linked with the hospital, which is the Queen Elizabeth Hospital down in Adelaide. And my team is in close collaboration with surgeons, the upper GI research, surgical team at the Queen Elizabeth. So we deal a lot with bowel cancers, patients that have liver metastasis. So bowel cancer, liver metastasis is the most common site for metastasis for bowel cancer, essentially, so the liver. About 50% of bowel cancer patients will develop this at a certain point in their stage, at the current rates anyway. Now, when you have liver metastatic bowel cancer, your survival rate drops really quite dramatically to 15% survival. And even if you have curative treatment, so in case for this type of cancer, it's usually through surgery. So the surgeon has to remove the tumor physically via resection. A lot of the patients normally aren't eligible for this treatment because of how the metastasis has occurred. So usually they will have to undergo a lot of treatments prior, and even still only a small proportion of the patients will be actually eventually eligible for what we call curative liver resection. And even within that, the 10-year survival rate is still quite low, 25%. So I think the key message here is like for the patients who are unfortunate enough to have received a diagnosis such as this, where it's quite advanced, it's really critical to find new treatments for these patients. And as people would have mentioned in this session, essentially, immunotherapy has been it's a game changer essentially for a lot of cancers. Now, in bowel cancer, that is true, but only for a very rare subset of patients at the moment. So usually when we think of immunotherapy, in the clinic, at least in the clinical sense, it's usually the checkpoint inhibitor blockade against PD-1 and CTLA-4. And this treatment actually works for a very small subset of bowel cancer patients called MSI patients, so microsatellite unstable patients. Those patients are less than 10% of the total bowel cancer cohort. So at the moment, there's still a massive proportion of patient cohorts that would really need new types of therapies. Other therapies being developed are cancer vaccines, which essentially stimulate the immune response, making, as Ben said, making cold tumors hot. And in bowel cancer, that's not as advanced yet, so it's not really in the clinic. So the next stage that are currently available for patients are adoptive cell therapies, where we actually expand our immune cell subsets in the laboratory and reinfuse it into the patient. Obviously, CAR T cells are, I think, the most commonly heard ones at the moment in the media. And so I won't be talking about that. But I'm talking about a specific cell subset called cytokine-induced killer cells, which is my specific interest for this Cure Cancer grant. So what are cytokine-induced killer cells? So they're a unique T cell subset where they're quite rare normally in the patient blood or even in tissue. And really they rely on the in vivo, ex vivo expansion in the laboratory to get large amounts of them. So this therapy is quite heterogeneous. And I think what people accept as the main mechanism of function for these cells is through their expression of both MHC-restricted and unrestricted recognition for cancer antigens. So for the non-immunologists out there, what this basically means is that we have both antigen specificity that's normally found in most of your T cells, but also non-antigen specific mechanisms such as antibody-dependent cell cytotoxicity using the CD16 antigen. I'm not sure if I can show a pointer. I'll just a pointer, I think you can see it. So ADCC, but also the second mechanism for tumor recognition, which is actually quite important for these cells, are through NK ligands and through the NK receptors. Now, once these cells are activated, they act like a normal CTL in terms of they are able to actually kill cancer cells through different mechanisms such as the granzyme preferentially release, induction of a pro-inflammatory microenvironment through interferon gamma, and also cell-mediated cytotoxicity through the TNF family, such as TNF alpha and the fast ligand pathways. So what is the clinical relevance of this in bowel cancer? So in bowel cancer, the reason why we were attracted to this is twofold. Firstly, coming from a small laboratory who aren't really specialized yet in cell manufacturing, the production methods to actually generate these cells seem quite achievable for us. There was quite a low adoption cost. And like I said before, because of the TCR or antigen independent mechanisms, it was quite attractive for us because it means that if we generate these cells, we could, in theory, implement it in our clinical treatment strategies immediately. And there was a paper that came out around when we first started looking into this therapy that suggested that in metastatic bowel cancer patients, if you give this therapy to these patients in conjunction with their standard chemotherapy treatment, you can prolong their survival rate from 2 years on average to 5 years. So it's not curative at the moment, but it still prolongs it by almost a doubling of the survival time. And we have a good oncology team that we collaborate with. So we asked them, like, if we can say to these patients that we could potentially give them 5 years instead of 2 years, would they take it? And is it worth it for us to develop further? And they said yes. And that's why we proceeded with this area of research. So the first thing that we've done really is we've just done a systematic review. And basically, that was just one report. We just wanted to figure out, is this actually true for a lot of colorectal cancer patient samples? Because there have been a lot of small clinical trials that have been reported around the world about this therapy. And the work that did this primarily was my PhD student at the time, who now has received her PhD, Celine, and also a medical oncologist, who's Yoko Tomita. And we've linked the paper here so you guys can look at it. But I think the key takeaway from it is, like, based from our systematic review, CIK therapy in conjunction with standard chemotherapy does improve overall and progression-free survival for patients. And another thing that's really important is, compared to other immunotherapies, there doesn't seem to have a lot of reported side effects. So it seemed to have quite low toxicity. So really, the results from this systematic review really motivated us to, let's try and progress this towards a clinic here in South Australia. And that's what we're trying, what our goal is at the moment, is to develop a CIK therapy protocol that we can give to SA metastatic CRC patients. So we have a published paper of how we think we're going to do this, and a lot of the research that we're doing will be based on this protocol and developing it to a point where it will be ready for the clinic. So the idea behind is, like, we identify patients suitable for our study, which is going to be first-line metastatic bowel cancer patients, phase 1 trial, where we collect PBMCs to generate our CIK cells, validate the CIK cells have differentiated into the cells that we want them to look like, but at the same time, see if we can get biopsies from these patients so we can grow patient-derived tumor organoids. And in the laboratory, make sure that our CIK cells can actually indeed eliminate the patient-derived tumor organoids from these patients as a way for us to confirm efficacy potentially when we reinfuse these patients. And then we have obviously standard things that you need for a cell therapy product. So sterility testing that we're going to explore, and also how to store them for multiple infusions for these patients. So to this end, we do have actually published a paper on our progress towards this therapy. In this scenario here, we just basically generated CIK cells from liver metastatic CRC patients. And again, this has been published work, so I won't really show the data, but I'll show the key takeaway message for this. One is that we can produce CIK cells in a very similar way to what's been published in the literature. These CIK cells that we've generated have the same molecular features, such as the NK receptors on these cells, cytokines being produced, and they also have same anti-cancer activity for both patient-derived organoids, but also we've used cancer cell lines both in 2D and 3D models. Now, what's important here and it's really what the Cure Cancer is funding, is we've not only seen like positive things about this therapy, but we've also seen things that where we think we can improve this therapy, potentially from just extending a patient's life to hopefully one day becoming curative. And one of the things that we have seen is the variability of the cell types being produced, which is probably not unexpected because it is a heterogeneous cell population, as I said before. And this heterogeneity also leads to differences in the cytotoxic activity of each product that we produce per patient. So for our Cure Cancer grant, we really wanted to ask the question, what makes CIK cells heterogeneous? So like, what are the populations present in this cell therapy? So when you look at the literature, there's no guidelines at the moment to say that this is a good CIK therapy product, except for the expansion of these CD3, CD56 double positive NK-like T cells. This is the data coming from a day 21 culture. So basically, CIK cells are presumed ready for a patient between day 14 to day 21 of our production process. And the criteria, like I said before, is we get from a really rare population. If you look at the PBMCs of a patient, this will be around 4 or 5% of your patient blood, essentially, to now about at least 40%, 30 to 40%, to sometimes even greater than that. So this is just a basic Tisney plot where we're just looking at a few markers, really, not just CD3, CD56, but other markers included in. But what you can appreciate is over time, it seems like they are clustering quite differently, even within the CD3, CD56, NK double positive population, which is indicated in blue here. So that suggests that indeed, even the ones that we're calling or we're using for our quality control, there might be different subsets already inside these in this therapy. So we've done a simple single-cell RNA sequencing pilot trial using one colorectal cancer patient and also one healthy control. And we've done some clustering and looking at the profiling of these cells. So this is still preliminary data, so we're not 100% sure with this data set, but we can confirm that we have found 9 unique subsets or clusters potentially. We confirm that in our single-cell sequencing data that our cell therapy product, the CIK cells, are all CD3 T cells, essentially. Most of them are CD8 T cells with some CD4 cells, and they express the other NK receptor, NKp30, which is marked as NCR3 here, just gene expression. And like I said before, we identified unique molecular signatures among the 9 clusters, as shown here. What's important is we identified the NK-like T cells, as shown by expression of both NK markers and also CTL markers. And also what's interesting is we found some cell subsets that have the proliferation gene. So we're calling these as the intermediary subsets, which might be important. So really what this data is highlighting is that there are opportunities to improve CIK cell therapy, which is what the Cure Cancer Grant is funding. We want to really understand what are the cells present in this therapy and identify the subsets that are really important, not only in killing the cells, but potentially in the future, figuring out which cells are important for long-term survival inside the patient that might be responsible for maintained cancer immunosurveillance. And secondly, what we've also shown is that potentially each patient, obviously we're not saying a complete comparison between healthy and CRC because this is an N equals 1 experiment, but we are seeing differences in proportion. So basically heterogeneity within the subcell composition in the culture that may also let us know why sometimes this is not as effective as other batches that we make, because we make this on a per patient basis because it's an autologous cell therapy. So I think I've touched about this already. So like aim 1 for us really is to just validate our single cell sequencing data. So we're going to try to expand our cohort by recruiting 3 more colorectal cancer patients and potentially 3 more healthy donors. So this is what's currently in progress for this year. Secondly, hopefully within the next few months, we'll finish the single cell sequencing. We'll identify markers that we can use to generate flow cytometry panels, so to potentially identify on a larger cohort of colorectal cancer patients what are their CIK compositions based on this new classification. So we are generating on a larger cohort around at least 10 to 15 patients worth CIK cells with matched patient-derived organoids. So these are liver metastatic patient samples that we're going to collect. and we're going to validate our findings. So we're going to try to make sure, do some cell sorting of our cell subsets that we found, see which ones are most effective in cancer killing, etc. So really the expected outcomes for this is to identify the effector cell subsets and intermediaries essential for CIK therapy-mediated anti-cancer activity. And secondly, by generating this cellular atlas, essentially, we hope to find cellular targets and molecular targets such as transcription factors that may be important to enhance these effector CIK cell subsets to generate new ways of producing the more effective cell subsets more consistently with higher efficacy. So ultimately, we are progressing towards a CIK therapy as a treatment option in metastatic colorectal cancer patients in Australia. And we're also, as I said before, identifying ways to improve the efficacy. So thank you for listening. So like I said before, I'm part of a larger team essentially, but I do lead the group for this research work. Our group leaders are both clinicians. So I work primarily with Professor Guy Maddern, who's a surgeon, upper GI surgeon here at the Queen Elizabeth Hospital, but also in close collaboration with Professor Tim Price, who's a medical oncologist. And I'd like also thank our consumers who have joined this team, our team last year, who's helping us throughout. So, and also finally, thank Your Cancer again for the funding.